All right, so today we'll continue on with our discussion in chapter 27 about these really strange group of organisms, the prokaryotes. One of the things that we keep in mind about the prokaryotes, good morning, Marie. One of the things we have to keep in mind about prokaryotic organisms, whether they're in the domain bacteria or archaea, is the fact that, as I mentioned before, these kinds of organisms do very well in extreme environments, radiation, pH, really warm temperatures, or very salty conditions. So we say that bacteria or prokaryotes are ubiquitous. They're all over the world or spread everywhere. You've got so many bacteria growing on your skin, in your bodies. One of the things we've got to be careful about is we don't get this germophobic, this germophobic idea that that there are germs everywhere. Keep in mind, of course, most of the bacteria that we hear about, or most of the prokaryotes we hear about, are those that are problematic, right, that cause disease, or perhaps even helpful. But a wide majority of prokaryotic organisms neither interact with us in any way. They're not harmful, they're not helpful, they pretty much do their thing. And in many cases, they're helpful to organisms in the environment. So before we start thinking that all bacteria or all prokaryotes are bad, you got to keep in mind that there's quite a few of them, or the wide majority of them, that are not. Then again, what happens is those that do cause problems are the ones that tend to get the most press. The interesting thing about this extreme environments that they can survive in, the radiation and the pH and the temperatures, gives a lot of credence to the idea, or at least a lot of support to the hypothesis, that prokaryotic organisms may have had the ability to travel through interstellar space, right? This is what I showed you last time, what we believe is a fossil, or could be a fossil of a prokaryotic critter. But remember, this was actually collected off of a rock or a meteor that we found in Antarctica that geologists tell us actually traveled from Mars. So if indeed this fossilized organism, or this structure is indeed a fossilized prokaryote, that would seem to indicate that not only are there perhaps or were living organisms on Mars, but then in many cases perhaps they can even travel across space in asteroids or meteors because of the fact that they can deal with such extreme conditions. I mean, the biggest problem in space, of course, is going to be, or one of the biggest problems is going to be radiation. And as I showed you before, they can stand incredible amounts of radiation, 3 million rads, with 3,000 times greater than a dose of radiation that would kill you. So prokaryotes, of course, we said, are typically unicellular, although they do tend to, and sometimes form together in colonies and even divide up labor amongst those colonies. And what's interesting about that is this is sort of a beginning of a story that we're gonna continue to talk about when we get into animals. And we talk about the collections of cells in animals that we call tissues. Now, we use the word tissues kind of loosely. Tissues is a collection of cells that performs a certain function, but it's in the animals that this idea of tissues is really raised to the next level. You think about the types of tissue that are contained in your body. You got nervous tissue, muscular tissue, cardiac tissue. Those are all very specialized tissue types. And while prokaryotes don't make tissues, and if you remember back from Bio 1 in the levels of organization, it's cells that make up tissues. Now, prokaryotes, a collection of prokaryotes is not considered a tissue because tissues have a very specific definition. They're a very specific structural and functional unit. But nonetheless, we do see even back in these organisms, not back in these organisms, but even amongst these relatively more primitive type of organisms, we do see this tendency, for at times anyway, for these cells to bond together in colonies. So that's something that's an interesting observation, because it just seems to be a trend that continues on as we move our way into the other groups of animals. As far as shapes goes, morphology, prokaryotes are pretty simple. They're either round or rod shape or spiral shaped. I did break the round shapes up into a little bit further subdivisions depending on how many are together. If it's two of them, it's called a diplococci. If they're in chains, a streptococci. So if you have strep throat, the organism that is causing strep throat is a streptococcus. So under the microscope, if we were looking at it, what we would see would be a chain of circular cells, circular shaped cells. And then staph is the one we said was like a grape, like a bunch of grapes, grape-like clusters. All right, so you can have a single round one, You can have 2 round ones, you can have a string of round ones, or you can have a group of round ones, all the round ones being called the cocci. They're not very big, of course. We don't often see, are often not able to see prokaryotes with the naked eye. The one exception is this one right here, the Thiomargarita nambiensis. Thiomargarita nambiensis is about 3/4 of a millimeter. Now, that's not very big, 750 microns, but it's still big enough that if you looked at it with just a simple, wouldn't even have to use a compound light microscope, but like a dissecting scope or a really good magnifying lens, you might be able to see. Right, there it is right there to kind of show you comparison compared to the bee. Typically, though, they're very small, 1 to 5 microns, compared to our cells, which are anywhere from 10 to 20 times larger than that. Prokaryotic organisms all possess a cell wall. Nearly all of them possess a cell wall, and the cell wall and surface structures too. And the cell wall in prokaryotic cells provides a lot of the same functions that we talked about back in Bio 1 in the plant cells, right? It maintains the shape of the cell. It provides physical protection. It protects a skeletal, quote unquote, a skeletal type of structure. Remember, plants don't have skeletons, right? So how do they stand up against the force of gravity? Well, it's because they have that cellulose, right? That carbohydrate, that cellulose material that's laid down in a fibrous network along their cell walls. I always think of it as kind of like the way you do fiberglass. Fiberglass is a series of strands of glass, fibers of glass, that are arranged in many different orientations. And then you just pour the resin over top of it to make a solid structure. The reason why fiberglass is lightweight and durable is it's made of glass, so that's relatively light, and it's durable because those glass fibers, the way they extend through, are all in a crosshatch pattern. You can imagine if you had a bunch of fibers that were all parallel, like that. Here we are again. Sorry about that. I lost connection for a second. But again, if those glass fibers were laid down in parallel fashions, it'd be real easy to break them along the grain, wouldn't they? So that crosshatch pattern, like the cellulose in plant cells, allows them to have some structural integrity. Well, the cell wall of prokaryotes is not made up of cellulose, but again, it provides the same sort of idea: cell shape, physical protection. And again, and this is very much like plant cells, it prevents from lysing in a hypotonic environment. Recall now, hypotonic environments is when water is going to move into a cell. And if we look at plant cells again, plant cells have that big central vacuole, and that central vacuole is used for storing water. So you remember, a lot of times when we're looking at a plant cell under a microscope, most of what we see is that central vacuole, especially if that cell's well hydrated. And this is all full of water. And in an animal cell, absorbing that much water could be problematic because animal cells don't have a cell wall. They might rupture, but that plant cell wall prevents the plant cells from rupturing. Another good thing for plants, because once again, they have a little different challenge when it comes to water. They can't go get water when they're dehydrated. Water has to come to them. Animals, of course, can move on down to the next stream or next lake to get a drink of water. All right, and I was talking toward the end of the lecture the last time about the differences in the cell walls between different types of prokaryotes. First, specifically the gram-positive and the gram-negative bacteria. Again, the gram-positive and gram-negative refers to a staining technique, which is what we'll get into today. And if you stain bacteria and they come back purple, then they're called gram-positive, and you can see in the picture down below, the ones down below are gram-negative, the ones that are red. And I'll show you during the during the staining technique why, because of the cell wall, why gram-positive bacteria are purple and why the other ones are red. We'll get that in just a minute, but just let me remind you again, gram-positive bacteria have a plasma membrane, well, as well as gram-negative bacteria. Gram-negative bacteria actually have two plasma membranes. In between is this special material that's only found in prokaryotes called peptidoglycan. And gram-positive bacteria have a very thick peptidoglycan layer on the outside, and gram-negative bacteria have a thin layer that's sandwiched in between those two layers of cell membranes. And many times gram-negative bacteria are the ones that we do hear a lot of the negative aspects about, because many of them or some of them can be disease causing. Oftentimes the symptoms of the disease is caused by an endotoxin, and a lot of gram-negative bacteria have these little green, you can see in the picture, little green, what do you want to call them? It's like little roots sticking up, shoots of a plant. Those are lipopolysaccharides, and those are what produce the endotoxins that are the symptoms of things like, for example, tetanus that I mentioned the other day. All right, let's talk about the basis for gram-positive and gram-negative bacteria. Okay, again, the gram is a stain technique. So let's see how that stain technique works about. So we're going to go from the left to the right, obviously. So the first thing we do is we apply a stain crystal violet to the bacteria in our sample. Crystal violet has a violet or a purple color. Crystal violet is a very wide range stain. We have some in the lab there at school. So we stain all bacteria purple. All right. So in this particular case, when you stain them all with crystal violet, the gram-positive and the gram-negative both become purple. All right. So the next thing we do is we come up with something called Gram's iodine, and we apply the Gram's iodine to it. And what the Gram's iodine does is it forms a complex between the dye, the violet, the crystal violet, and the iodine. It forms a complex inside the cells. In other words, it locks the stain in. So it makes the stain more permanent, I guess is a good way of saying it. Now, the third step, this is very important. In the third step, alcohol is applied. Now, if you remember back in Bio 1, alcohol is a drying agent. It dries things out. So in the gram-positive bacteria, step back one more second, gram-positive bacteria that have that thick peptidoglycan layer, when you apply alcohol to a peptidoglycan layer, it shrinks. It dehydrates, so it shrivels down. And in gram-positive bacteria, when you apply that alcohol, that shrinking of the PG layer that you see there, the peptidoglycan layer, traps the dye inside of the bacteria. So it provides an impenetrable barrier keeping the dye inside. But on the other hand, the gram-negative bacteria, remember, they have a very thin peptidoglycan layer. So when the alcohol dehydrates and shrinks it, because it's so thin, it doesn't provide that impenetrable barrier. So the alcohol shrinks the peptidoglycan layer a little bit, but not enough to prevent the stain from leaching out. So that seems kind of funny. You stain them, then you've unstained them. Well, you notice in panel 3, we've already got a difference between the two. After the alcohol, the gram-positives stay purple, yeah, and the gram-negatives lose the purple dye. Now, then we follow up in the fourth step with something called saffron. Saffron is a red dye, and we apply that to all of the bacteria again. Now, the ones that are dark purple are going to still stay dark purple. They take up the dye too, but the dark purple dye masks the red dye, so they're going to look purple. But the gram-negative ones, which lost the dye in step 3, they're going to take on the saffron red dye as well, and since there's no other dye there, they're going to look red. All right, so that's the basis of bacteria being either gram-positive or gram-negative. You can see in the picture down below, quite clearly there's a number of gram-positive and gram-negative bacteria in there, right? The gram-positive ones are purple, gram-negative ones are red, and in a little bit of picture, right, you can not only tell gram-positive and gram-negative here, but if you look real closely, you can tell shapes too. It looks to me like the gram-positive, for the most part, are cocci. They're all round-shaped. And a lot of the gram-negatives, as I'm looking through here, look like they're all bacilli or rod-shaped. Now, the reason why this is so important for microbiologists is that, for example, if we're trying to diagnose a bacterial infection, if we suspect that the patient has a bacterial infection caused by a gram-negative bacteria, this is how we would do it. We would take a sample from the infected individual. We would do a gram stain, and if they came back red, then that would support our idea that these are gram-negative bacteria. Right? Very simple staining technique. If you take microbiology, you'll learn how to do a gram stain. That's the basis behind gram positive and gram negative. And just remember, when we're identifying gram positive or purple, gram negative or not. That's how I remember. Gram positive, purple, they both start with the same letter. All right, now, not only do prokaryotic organisms have cell walls, but they also have surface structures. They also have structures on the outside of the cell wall that provide a certain function. Like, for example, here is Streptococcus, the organism that causes strep throat. They've labeled the bacterial cell wall that you see there, but notice on the outside, the part that's in the green, that's a bacterial capsule. Now, some bacteria have a bacterial capsule, some have a slime layer. Slime layers tend to be a little less dense than capsules, but either one, whether it's a capsule or a slime layer, these things have this structure on the outside, this bacterial surface structure has three basic functions, and I've listed those up above. One other function is adherence. So this is why the Streptococcus is sticking. Adherence means to stick to, like adhesive, to the tonsil cell. The capsule of the slime layer can also prevent them from drying out, so it's an anti-desiccant. All living organisms, whether they're prokaryotes or eukaryotes, are all affected by desiccation the same way. Some organisms can survive better being desiccated than others, but this is a good protection to keep these guys from drying out. And another thing they do is they hide them, these capsules and slime layers, they hide them from your immune system. What I mean by that is your immune system is a series of cells that goes around and checks for foreign cells. It checks for cells in your body that are not supposed to be there. It's not only just cells. Your immune system also responds to things like pollen as well. It's if you have allergies, that's what your body is doing with an allergic response. An allergic response, your body is responding to an allergen. Pollen, if you're allergic to it, is an allergen. Mold could be an allergen. Some folks are allergic to cats. The dandruff on the cat, not actually the hair, but it's the dandruff on the cat that's actually causing the allergic reaction. And your immune system, if you're someone who suffers from allergies, just kind of goes overboard, really. An allergic response is sort of an over response to a foreign particle. Pollen, cat dandruff, mold, in many cases, not necessarily with all mold, but in many cases, those things aren't going to cause death. But your immune system is attacking them still, and it acts like it needs to get rid of them. So that's why you get the runny nose and the sneezing and the coughing. You're trying to expel this allergen because your immune system thinks it is something bad. We said pollen's not going to hurt you, but your immune system is simple because it basically works on examining the structures of these foreign particles, the outside structures. So it's kind of a feeling thing. Immune systems go, remember in Bio 1 we talked about glycolipids, glycoproteins, those things that extend out from the surface of a cell. Your immune system feels those glycolipids and glycoproteins, and if they have the right shape, then everything is fine. I think in Bio 1 I mentioned to you that's the problem with transplanting, right? If you transplant a heart from a heart donor to the heart recipient, the immune system in the heart recipient sees the cells of the new heart as being foreign because the glycolipids and the glycoproteins, which the structures are genetically determined, they're from the genetics of the heart donor. Your immune system sees that as being foreign and tries to attack it, and that's why we have a problem with tissue rejection sometimes when we have transplants that go on. Folks who receive transplants will often receive immunosuppressant drugs, which basically kind of calm down the immune system, and with the hopes that eventually your immune system will become used to these new cells and accept them as not being foreign anymore, won't attack them. But if you're a bacterial cell and you've covered yourself with this capsule or a slime layer, then you're essentially hidden from your immune system. Your immune system can't feel any of the glycolipids and glycoproteins, so it assumes that essentially assumes it's not there. So it more or less makes them invisible, right? Not invisible literally, of course, but invisible to your immune system. Bacteria also have an interesting way of surviving environments that aren't conducive to their survival. Surviving environments that aren't conducive to their survival. In other words, if the environments are changing, and we learned enough about evolution that organism survival is all about the environment, right? So if the environment is not conducive to your survival, then organisms can go extinct. I mean, basically, organisms have to adapt to a changing environment, or they will go extinct, or at least if they don't go extinct, they will suffer a population crash. In certain organisms, if the environment changes, what they'll do is they can move, they can migrate to a place in which the environment is better. They can undergo adaptation as well. They could undergo natural selection, adaptation, and hopefully adapt to the environment. But of course, most of the time, as we've learned, what's going to happen to them is they go extinct. So some organisms have come up with a way to kind of beat the system, if you will. And what I mean by this is there are some species of bacteria or prokaryotes, and it doesn't occur in just prokaryotes, it also occurs in other species we'll talk about. But like this one here, this is Bacillus anthracis. This is the bacterial organism that causes anthrax. And what anthrax will do, or the organism that causes anthrax, is during periods in which the environment is not conducive for them, they'll form an endospore. So what will happen is this endospore will form, and then the rest of the cell will disintegrate, and that endospore will lay dormant. Dormancy is kind of a strange subject in biology. Endospores aren't dead, because you can't bring things back from the dead. They're not really alive. They're kind of in this suspended animation state. Think of it as kind of like hibernation, although when a bear hibernates, it's still alive, okay? But think of it as being sort of in a hibernation state. And what these endospores do is they're usually very resistant. They usually have outer coatings that are very resistant to the environment. And endospores don't survive the changing environment by adapting to it. They survive the changing environment by kind of hunkering down into this spore and waiting for the environment to change. And some of these endospores in these organisms can last quite a while, days, weeks, decades, centuries in some cases. So you see, it's kind of another alternative to a changing environment. The environment changes and you can't change with it, and you can't move, can't go anywhere. One of the things you can do is form an endospore and just kind of wait for the environment to change to become more conducive. So an endospore is another modification to the cell surfaces. Other modifications include things called fimbriae. Fimbriae are little projections that extend out from the cell surface that are used for adherence. So if we go back to our strep throat organism, they would use fimbriae to attach themselves to the tonsil cell as well as the slime layer. Fimbriae can also be used to exchange DNA. Now, prokaryotic organisms, their genetic diversity, the way they get to genetic diversity is going to be very different than the way we do. Most of our genetic diversity in us eukaryotes comes from sexual reproduction, right? The mixing of the sperm and the egg. You don't have that in prokaryotic organisms. There is no sexual reproduction in them. It's all asexual, which means they're clones of one another. If you remember back in evolution, the way that they get around the fact that every time you copy DNA, as I mentioned, you might get a little different DNA each time. And that doesn't occur at enough of a rate of success in eukaryotic cells to be a real influence in variation. You remember in the Hardy-Weinberg equilibrium, we said that allelic mutations aren't a significant contributor to eukaryotic cell variation or eukaryotic organism variation. What bacteria do, or prokaryotes do, is even though variation or allelic mutations is not a major source, they produce lots of offspring. So even though, many times when you change the DNA, it's actually a bad thing, if you produce lots of offspring, it's a numbers game. There's a better chance that you'll come up with the right allelic mutation that allows an organism to survive. But there is a way that prokaryotes can actually exchange DNA, and that's an extension of the fimbriae that grows between two of them. We call a pili, or a sex pili. The idea behind the sex, obviously these two organisms aren't having sex, but what they're doing is they're exchanging DNA between them, right? So that's another cellular structure on prokaryotes that can be used for a purpose. And we'll talk a little bit more about this whole idea of genetic information changing and being exchanged in just a little bit. All right, how do these guys get around? Well, motility occurs in about half of prokaryotic species. Some are actually pretty fast. Scale-wise, some of them can move up to 90 miles an hour. And they're not moving 90 miles an hour in our world, but if you were a prokaryote, the relative speed is moving up to 90 miles an hour. There are lots of predatory prokaryotes out there, and they're very fast. That's moving pretty quick, 90 miles an hour, again, at that scale. The typical methods of mobility include a flagella, which is the most common, not like a eukaryotic flagella, right? The flagella we find in prokaryotes is different than in eukaryotes, different than we might find in, like, say, a euglena. So here's a cross-section of what a prokaryotic flagella looks like. And again, down in the lower right, I borrowed a picture from another source to kind of remind you what eukaryotic flagella are a series of microtubules that bond together, and the way that flagella move back and forth is those microtubules move up and down against each other, and they cause the flagella to wave back and forth. So up, down, right? See what I mean? In the case of the prokaryotic, it's more a matter of spinning. They actually have a rotor, a very interesting rotor that extends. This is obviously, we're looking at gram-negative since we have the two cell membranes there. But that spinning action is what moves the flagella in this case. Notice down below here I wrote the word exaptation. You remember how we mentioned that an exaptation is like eyes, feathers, legs back in evolution? We said an exaptation was a characteristic that arose for one purpose that became fortuitous for another. The reason why I'm indicating this as exaptation is what makes this flagella spin, what causes the rod portion of the flagella to spin around, is the movement of protons from one side of the membrane to the other. So you see these red arrows are indicating that protons are flowing through this region, and the flowing of protons is spinning the rotor. Now the reason why that's an exaptation is, if you remember a little bit later on when you talked about respiration and photosynthesis in Bio 1, you remember ATP synthase? ATP synthase worked the same way, didn't it? The whole purpose, or one of the major functions that's going on in respiration is the movement of protons from one side of the membrane to the other. It also happens during the light reactions of the photosynthesis. And when you get more protons on one side, Mother Nature takes over and says, oh, that's disequilibrium. So those protons flow back to the other side. And when they flow through ATP synthase, they turn the catalytic knobs of the ATP synthase to make ATP. In this case, the protons flowing through spin the rod around to turn the flagella. This is a great example of convergent evolution too, isn't it? Right? These two structures, the flagella in a prokaryote and the flagella in a eukaryote, are very different in the structures that they are, but they have the same function. Right? So it's kind of like the wings of the insect and the wings of a bird. Right? Those wings are very different structures, and they arose from very different adaptations through time, but they perform the same function. So even down here at the very low microscopic level, we see convergent evolution. I always find this fascinating, and folks that are into nanotechnology is the technology of building really small things. Nanotechnologists get fascinated by this type of stuff because this is basically a spinning wheel. So if we could somehow at the microscopic level mimic this, we could make miniature spinning wheels and gears and make miniature devices and miniature equipment, or miniature things, right? So we look back to the prokaryotes and we find out, these guys have already come up with it. We just kind of borrow from Mother Nature. All right. Now, as far as motility goes amongst these prokaryotic cells, we involve the word taxis to explain their movement. Taxis is to move towards or away from a stimuli. If you think about, maybe that's where the word taxi came from, right? Taxi moves you from one place to the other. And you can be either chemotaxic, which means that you're moving towards or away from a stimulus. In this case, the stimulus is environmental chemicals. So prokaryotic organisms could move towards or away from certain environmental chemicals. If you're a photosynthetic species of prokaryote, being able to respond to phototaxis would be really important, right? They respond to light. And there are even some that respond to magnetic fields. We call those geotaxis. And if you think, why would prokaryotes care about the magnetic field on Earth? Well, if you think about those that live in a lake, the nutrient-rich part of the lake is at the bottom. And being able to know the top of the lake and the bottom of the lake, and you guys don't have eyes or anything like that, is important, because the nutrients that the bacteria need are at the bottom of the lake, and being able to sense magnetic fields lets them sense where's the top of the lake and the bottom of the lake. All right? So chemotaxis, phototaxis, and geotaxis. All ways in which these organisms move to or away from things. Using, in many cases, flagella. Some use slimy threads, and they kind of pull themselves along. But either way, it's motility. All right. Now, when we were in Bio 1 and you talked about the different parts of the cell, there were quite a few, right? There was a nucleus, ribosomes, an endoplasmic reticulum. Well, it's relatively simpler in prokaryotic cells. For one thing, they don't have a nucleus, right? They're anuclear. They have a nucleoid region. They do have a chromosome. It's one, right? One single loop chromosome. There's no internal compartmentalization. And what I mean by that, if you remember a eukaryotic cell, all of the organelles, like let's say a mitochondria, have a membrane around them, right? Even the nucleus has its own nuclear membrane around it. So cells are compartmentalized inside a cell compartment, right? So you get that the cell, which is the compartmentalization, and then each of the organelles is compartmentalized. Don't know if you remember, but back in Bio 1, we talked about peroxisomes. Peroxisomes are responsible for getting rid of hydrogen peroxide. When your body digests fats, one of the byproducts is hydrogen peroxide, and hydrogen peroxide is actually a very strong oxidizer. It's not good for cells. So when your peroxisomes, you break down that hydrogen peroxide, use an enzyme called catalase by which to do that. And at the time when we talked about in Bio 1, we said that this internal compartmentalization allows peroxisomes to contain that hydrogen peroxide inside the cell without affecting the other parts of the cell. So that stuff is contained within the compartmentalization of a peroxisome. Well, we don't see that in prokaryotic cells, but we do see infolding, all right? Infolding of the membranes, not internal compartmentalization, but infolding. And infolding is very important because infolding forms membranes. And you remember, back to Bio 1, respiration in the mitochondria, and then, of course, the light reactions that occur in photosynthetic cells in the chloroplast are all done across membranes. Membranes are crucial to those two functions, to respiration and photosynthesis. Remember, respiration and the light reactions of photosynthesis all depend on forming a disequilibrium. Just like I was talking about with the flagella a second, the whole idea is to make a situation where you have more protons on one side than the other. You remember in respiration, you can't get protons flowing through that ATP synthase that I just mentioned unless you have more on one side than the other. That's just the law of physics. So you got to have membranes. In prokaryotic cells, there's no mitochondria, there's no chloroplasts. Remember our endosymbiotic theory, we think that chloroplasts and mitochondria were actually prokaryotes at one time. But prokaryotes can form infolding membranes they can use in respiration. So in the one on the left, you see a respiratory membrane. And then photosynthetic prokaryotes, they can infold their membranes and form the membranes necessary, like in this case, a thylakoid membrane to form photosynthesis, right? So you got to have that membrane. And in prokaryotic cells, since they don't have that compartmentalization, they can infold their membranes or infold the cell membrane and form these respiratory thylakoid membranes. As I mentioned, they don't have nearly as much genetic information. They have a relatively simple genome, that collection of genetic information, about 1,000th as much DNA as we do. One of the reasons that is because, for one thing, prokaryotic cells do not have introns. They only have the exons, the only expressed part. Remember, we have a lot of maybe what you might call junk DNA in our DNA. That's the introns, the parts that's not read. Prokaryotes, for the most part, most prokaryotes don't have the introns, so they have less DNA. And also the fact that, they have a much simpler lifestyle than we do, so they're not going to need as much DNA information by which to do that. You can see down below, this is a bacterial cell in which they have sent it through a process, and it has spit out or expelled its chromosome, and all these little winding, little loops that you see here, those are all that one chromosome. If we unwound all of those, remember, chromosomes occur all in a single loop. So if we undid all those windings and pulled that apart, there would still be one chromosome all in one singular loop. Now, one thing that prokaryotic cells have that eukaryotics do not is they also, in many cases, have accessory loops of DNA we call plasmids. Little chunks right here. These are pieces of DNA outside of the main DNA. And plasmids are interesting among prokaryotic cells because they can provide for things like antibiotic resistance, special contingencies, the ability to digest certain foods in different types of environments. Plasmids can replicate independent of the main chromosome, and they can be passed to one another. Like, for example, in that picture of the sex pili that I showed you, there we go. You can pass plasmids between sex pili. Now, what this means from a health point of view or from a medical point of view is if a bacterial organism or prokaryotic organism has developed an antibiotic resistance, and it's developed that antibiotic resistance because of some information in its plasmids, getting together with another prokaryote, forming that sex pili, one prokaryotic organism can pass that plasmid genetic information, a copy of it anyway, to the other one, and now that other cell has the antibiotic resistance. So it's a little different than what we talked about with Darwin, right? With natural selection, where, like, the reason why these organisms become antibiotic resistant is because they produce offspring that survive better. Well, that does happen, but another way they can do it is two prokaryotic organisms can get together, and if this one prokaryotic organism doesn't have the resistance to the antibiotic and this other one does, they can get together, make a copy, and pass that plasmid to that one that didn't have antibiotic resistance before, and then all of a sudden now it's resistant. It's one of the problems dealing with pathogenic prokaryotes is this ability to pass plasmids back and forth. I always think that's kind of cool. It'd be kind of neat if you could absorb DNA from other organisms, right? I think I don't know if I mentioned you guys, but usually Bio 2, I said if I could do anything that another animal could do, in spite of the fact how much I love the ocean and fish and sharks and all that, I think it would be flying, to be honest with you. I wish it'd be kind of cool to be able to fly. So it'd be neat to be able to absorb some bird DNA and be able to fly. Now eukaryotic cells don't have plasmids, so I wouldn't be able to do it to a plasmid. And what I'm talking about is virtually impossible, but it is kind of the same thing. So if you got two bacteria, and one is got an antibiotic resistance, and another bacteria is like, oh my gosh, this antibiotic, I can't handle, I can't deal with it. It's killing me off here. One bacteria can say, well, listen, I'll pass you a copy of my plasmid, and now guess what? Because of the genetic information in that plasmid, you are now resistant to that antibiotic. It's kind of freaky if you think about it in a way, right? It's kind of freaky if you think about bacteria getting together and passing ways to deal with the environment, whether be these special contingencies of digesting certain types of organic material or antibiotic resistance. Not so good for us, right? So if you kill them off, the ones you don't kill off who have the plasmids, the ones that through reproduction occur after that, can then be able to have antibiotic resistance because you can pass plasmids between them. Sneaky little things, those prokaryotes. All right, genetic variability. Mutations arise spontaneously in bacteria. They arise spontaneously in eukaryotic cells as well, and that occurs through radiation and through chemicals. And just through the fact that, like I said, when you copy DNA, it doesn't get copied the same way every time. There can be mistakes. Now, many times there are provisions to correct those mistakes, but sometimes mistakes slip through, and many times, most times, those mistakes are deadly, right? But sometimes they're beneficial. How do prokaryotic cells get away with the fact that most of the time when you have mutations, it's going to be something bad? Well, again, they do it by reproduction. The amount of cells that are produced from a prokaryotic cell, even in a day, can be upwards of hundreds or thousands or millions. So it's rolling the dice. If you know that these genetic mutations are going to occur, and you know that 1 out of 1,000,000 is going to be beneficial, and if you make 1,000,000 or 10 million offspring, then you've created 10 organisms that have a beneficial mutation. So prokaryotes, because they have this rapid ability to reproduce through asexual reproduction, right? Boop, 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 they just divide and keep on going. These organisms can deal with these kinds of spontaneous mutations that, for the most part, in us eukaryotes would be deadly. Mutations, of course, into the DNA and also plasmids that we just talked about can be very quickly spread through a population, too. We hear a lot recently about MRSA. MRSA is Staphylococcus aureus that is resistant to an antibiotic known as methicillin. Methicillin-resistant Staphylococcus aureus, or MRSA. Staphylococcus aureus is a major prokaryotic problem, especially in hospitals. Hospitals are always dealing with Staphylococcus aureus infections or staph infections. And one of the ways that they can deal with it is by using methicillin, an antibiotic. But as time has gone on, what we have done is actually created strains of Staph aureus that are MRSA, that are methicillin resistant. And they do that through mutations and also through passing plasmids. So if one of those organisms had a plasmid that made it resistant to methicillin, it could just pass that plasmid, just like we see there. It could pass that plasmid to another one. There's no meiosis in these organisms, so it's all horizontal gene transfer. There's no vertical gene transfer. Vertical gene transfer, again, is transfers of information from parents to offspring, offspring to their offspring. So you're moving down. Horizontal, this would be an example of horizontal gene transfer. You're passing it between two, for lack of a better way, two adult organisms. Genetic variability among prokaryotes can occur in 3 major ways. Transformation is one way. Conjugation is another way. What you're seeing there in the picture on the right is conjugation, where one cell transfers genes to another. And then genetic variability can be increased or can be spread amongst prokaryotic organisms by use of a vector. A vector is anything that spreads something. We often think about vectors in terms of disease, like malaria. Malaria is spread by a mosquito. Malaria, the disease, is actually caused by a protozoan. The protozoan is spread from victim to victim through the mosquito, so the mosquito is a vector. In the case of transduction, the vector in this case is the virus. The virus can transfer genetic information between bacterial organisms, increasing genetic variability. Let's take a look at transformation first. Transformation, if you remember, that's what Griffith was the guy in Bio 1 that we talked about with the mice. Remember, he injected the mice with the living R and the living S, and the living R didn't kill the mice, but the living S did. So then he takes the S and he kills it, and he injects into the mouse the living R, which won't kill the mouse, the dead S, which shouldn't kill the mouse, but indeed the mouse does die. And when he does a necropsy on the mouse, he finds that there's living S in the mouse. He didn't inject the mouse with living S strain of that bacteria. The living R was transformed. You see, one thing that prokaryotic organisms can do, if you're following the picture on the right, so a cell dies, right? bacterial cell dies, and it releases pieces of its DNA, the part in the blue. Bacterial cells can incorporate DNA from the environment. So the DNA from the dead cell can be picked up by another live cell, the part in the blue, and then incorporated into their DNA. So that's what we call natural transformation. That's what was going on. That's why those living S showed up in the necropsy of the mouse. Even though he didn't inject them, the living R took up the genetic information from the dead S strain of bacteria, and the living S was pathogenic, caused disease because of its DNA information. Well, now the living R has that DNA information, and now it's no longer the R strain, but it's the S strain, and it can cause death. We've done this in the lab too. It's called artificial transformation. We've used this ability to transform E. coli. E. coli, a prokaryotic organism, what we do is we can inject or place into the environment genetic information that the E. coli will take up. Specifically, the kind of artificial transformation we'd be interested in would be, say, the production of insulin, right? Insulin, which is necessary for diabetics. E. coli have no use for insulin, obviously, but if we could inject, the insulin gene into a bunch of E. coli, and then E. coli can reproduce like crazy because they reproduce asexually, as they read their genetic information, remember in their case, they don't have any introns, so they'll read it all. As they go through the part that's been incorporated, the part in the blue there, they'll start producing insulin. Now, since they have no use for insulin, the insulin just gets dumped or exported into the environment. So pharmaceutical companies that are producing insulin will have these big vats of E. coli that are growing with this insulin gene in them. And as the E. coli goes about its daily activity of using its genetic information to do all of its E. coli stuff, it, basically unwittingly produces insulin, and then we drain that off and use it for our own medical purposes. This is what we call molecular cloning when we transfer things like this, because we're actually cloning the genetic information and incorporating it into another organism. All right, so that's transformation. The other one was conjugation, right? Conjugation, remember, is when we get two cells together, like we have on the right with a sex pili. So let's take a look at how that works. There we go. All right, there's another picture of two bacterial cells doing it, I guess. So look in the picture up above. There's a plasmid. We'll call it an F plasmid. And if you contain that plasmid, then you're an F plus cell. And if you don't have that plasmid, you're an F minus. So that's what we're starting off with. So the purpose here is to transfer the genetic information in the F plasmid, which could be how to be resistant to methicillin or whatever special contingency you want to, is to transfer it to the F minus. Well, you don't want to just move the plasmid from one to the other, because then all you've done is switch who's the F plus and the F minus. So you notice the sex pili forms between them, and then this thing called rolling circle replication occurs. It's really kind of cool. The F plus cell only donates half of its DNA. You see the part that's going through the sex pili is half of the DNA, one strand. That's then passed into the F minus. Now, if you remember Chargaff's rule, adenine, guanine, cytosine, and thymine, that template, that half of strand that's being passed to the F minus cell, the F minus cell can just fill in the other half, right? Following Chargaff's rule. And then the strand that's being lost by the F plus cell, that can be replaced for the same reason, using Chargaff's rule. And so you can see the purple in the F plus is the part on the outside, and in the new F plus, the part that's been added new is the inside. So that plasmid now has been passed from one to the other. We call this conjugation because the two cells conjugate together through that sex pili, through a conjugation bridge or a sex pili. And this ends up in two F plus cells, or two cells that now have the plasmid. And they can do this pretty quickly too. So yeah, again, if you had the F plasmid that made you resistant to methicillin or some other antibiotic, you can quickly pass it on to the other one. That's why doctors now are a little less apt to freely prescribe antibiotics, because what you basically do is you basically, when you use antibiotics, in wide use of antibiotics, you're actually stimulating bacterial cells to engage in this process by which they can adapt. So you're actually selecting for those ones that are resistant to your antibiotic. And that's what happened with methicillin. Once enough methicillin had been used, we had unwittingly selected for methicillin-resistant Staph aureus. And so what that means is you have to switch to another antibiotic. So doctors now are a little less inclined to prescribe antibiotics to people, especially for low intensity infections, because your body will do a pretty good job of getting rid of that, right? It'll get rid of that bacterial organism for the most part. All right, the last way involves viruses. Now, I don't talk about viruses in Bio 2. Viruses are a weird little thing. I call them thing. They're not really alive. They have some of the characteristics of being alive, but not all of them. If you remember the characteristics, organization, metabolism, all that sort of stuff. For example, a virus can't reproduce by itself. It needs a host cell. That's what you're seeing in the picture there. The green is the host prokaryotic cell, and all those little orange spaceship-looking things in the upper right. I got a little diagram of these viruses. These things are called bacteriophages. These kinds of viruses. A bacteriophage eats. Phage is to eat. Eats or destroys bacteria. And what a virus does is a virus has its viral DNA, but like I said, it can't make copies of its DNA, it can't make copies of itself unless it has a host cell. It attaches itself. It's really kind of cool. It looks like this sort of bizarre spaceship kind of thing. In this case, in the picture in the upper right, the virus is attached to an E. coli, and then it injects that bacterial information into the host cell. And then let's see what happens after that. So we start up at the top. Here's our bacteriophage. And that phage injects a little bit of its phage, so that's the viral DNA, into the host cell. And in the host cell, we have a couple of alleles. Just for example, we have an A+ allele and a B+ allele, right? That's the donor cell. Now, what the phage DNA does, once it's injected inside of the cell, is viruses cause the host cell's DNA to break apart. And when it does that, it uses the machinery of the host cell, or in this case, they're calling it the donor cell. It uses the ribosomes and the machinery of the transcription and the translation inside that cell to make more viral particles. It makes the caps. It makes all of the strain structures that we see here. This is the cap, the part that injects the little legs that attach to it. All of those are proteins. The virus has the information on how to make those, but it doesn't have the ribosomes. So the little purple dots that you see in the donor cell, those are the ribosomes. So the phage DNA is replicated proteins, phage proteins are synthesized, and then all of that DNA information is repackaged into new viral particles. Now, some of the viral particles in the head of the viruses have just phage DNA, the part that's in the blue. But remember, I said in stage 2, the virus causes the host cells or the donor cells' DNA to break up into chunks as well. And sometimes pieces of that DNA get incorporated into a virus, like you see the one that's labeled A+. So the A+ allele ended up being crammed into that virus. Now that virus goes to another cell, which is an A-, B- cell, injects the A+ allele, and now you've got what we call a recombinant cell, right? Recombinant means that the DNA has been mixed up. During crossing over in meiosis, just to remind you, what you create are recombinant DNA. You're recombining the DNA in a different order, different structure. This is adding genetic variability to prokaryotic cells, because now that recipient cell, which used to have the A- and the B- alleles, now has an A+ and the B- allele. Right? So you mixed up the genetic information. So you see what I mean by a vector, right? The virus is the vector. It moved the phage DNA into another. And of course, then the recombinant cell can be attacked by a virus, and the whole process can be repeated again. So they can either pass DNA between themselves by picking up from the environment, They can pass or change their DNA by picking up pieces of DNA from their environment. They can change their DNA by transferring genetic information between two bacterial cells, or a virus can act as a vector for introducing genetic information into a cell. So that's how they get their genetic variability. We get our genetic variability, you remember, from most of our genetic variability from crossing over, which happens in meiosis, which you're not going to see in these organisms. All right, so enough about reproduction. Let's talk about how they make a living. Nutrition and metabolism. Now, I understand that this slide looks pretty busy. It looks busier than it really is because I've included some pictures to give you some examples. I would like you to know the different types and the different ways in which bacterial or prokaryotic organisms make a living. So if you're thinking of some type of a study guide, recreating this chart would be a good idea, but don't worry about the pictures, and you'll see when you take the pictures out, it's actually a little simpler. So nutrition and metabolism in all organisms on Earth, since we're a carbon-based life form, involve two things. You got to have a source of carbon. Okay, think about plants when they photosynthesize. Their source of carbon is carbon dioxide, right? Think about us. Our source of carbon is the food we eat. We eat other carbon-based life forms, and we use that carbon atom in our bodies to make other structures, right? So you need the building blocks to, if you're a carbon-based life form, you need those carbon building blocks to make structures in yourself. So when you eat food, you get two things out of food. You get the energy from the food, and you get the carbon in the food, which you then use to make carbon compounds in your own body, right? All those things you talked about in Bio 1. So let's start with the carbon source first. In other words, let's start with where do you get your carbon? And let's go to the left-hand side, because that's one you're maybe a little more familiar with, especially when you think about photosynthesis. If your carbon source is carbon dioxide, then we say you're an autotroph. All right, troph is to feed, okay? So plants are autotrophs. Now, what you didn't get into in Bio 1 is the fact that the difference among autotrophs can be where they get their energy. And again, let's go with the ones we're most familiar with. If you get your energy from light, then you're a photoautotroph. All right, the auto means that you use an inorganic material to get your carbon. Carbon dioxide is inorganic. Remember the definition of organic. Organic compounds contain carbon and hydrogen. Carbon dioxide's only got one of those. All right, so it uses the carbon dioxide and inorganic materials as source of carbon. And if you're a photoautotroph, you use light as your source of energy. And that's what most plants do. Right down here at the bottom, that's what plants do. Magnolia, the palm, mosses, that's what they do. Typical plant photosynthesis. There are other critters that do it that way. For example, there are some cyanobacteria, some blue-green algae, or some green sulfur bacteria, or purple sulfur bacteria. This is green sulfur bacteria. This is purple rhodobacter. Purple photosynthetic bacteria are able to photosynthesize by using a pigment slightly different than chlorophyll to absorb wavelengths of light. That's why they look purple. And there are even some protozoans like Euglena that can do that. They can photosynthesize. Now, the blue arrows, the dashed arrows that you see, I'll get to those in just a second, because what that indicates is showing you is that some organisms can actually make use of more than one way of doing this. All right, so those are the photoautotrophs. Light is the source of energy. Carbon dioxide is the source of carbon. Now, there is another way to get energy, not using light. When you oxidize something, this is going back to Bio 1 again, when you oxidize something, you release energy. And if you're oxidizing something that's inorganic, it's called inorganic oxidation. So those organisms that do that are called chemoautotrophs. All right, in the blue there, I have a little key to help you remember how the name works. So first you take the energy source, right? In inorganic oxidation, it's chemo. And then you take the carbon source, which is carbon dioxide, it's auto, so chemoautotrophs. Chemoautotrophs use carbon dioxide as their form of carbon, and they use the oxidation of chemical compounds as their form of energy. Now, chemoautotrophs are only found among prokaryotes. They're the only organisms that can do that. There are these organisms that are methanogens, for example. Methanogens take carbon dioxide and oxygen, carbon dioxide and hydrogen, and they make methane. And that process releases energy. All right, so that's the energy they need to build up carbon compounds. Purple sulfur bacteria, not purple photosynthetic, but purple sulfur bacteria can oxidize sulfur or hydrogen sulfide and release that energy and use it for their needs. Hydrogen sulfide is a very common gas that comes out of those volcanoes and those deep smoker vents that we were talking about. So there are even organisms around today that can make use of hydrogen sulfide for a source of energy. Nitrifying bacteria. Nitrifying bacteria use ammonia as a source. They oxidize ammonia. Nitrifying bacteria are very important because of the fact they're involved in ecology. Now, I'll show you a little bit of bigger picture of this a couple slides later, but nitrifying bacteria are those bacteria that are important for introducing nitrogen into the soil. Plants need nitrogen to survive. Your fertilizers contain nitrogen. Nitrogen is oftentimes the limiting resources in a plant's environment. So these nitrifying bacteria are very good at providing that nitrogen. Now, if you're thinking, well, wait a minute, the air is 80% nitrogen. Why can't plants use that? They can't use atmospheric nitrogen. These nitrifying bacteria also break down potentially toxic substances like ammonia, keeping the environment safe. All right. Now let's go to the other side. If where you get your carbon is not from carbon dioxide, but from something organic, then you're called a heterotroph. That would be us, right? And if you get your energy from the oxidation of these organic compounds, then you're a chemoheterotroph. And if the oxidation that you engage in is oxidation of an organic compound, then you're like us. You're like animals. Animals, fungi, some plants, many prokaryotes, Clostridium, for example. This is Clostridium. Clostridium can cause botulism. Botulism is one of the most deadly types of food poisoning out there. If you get a good dose of botulism, usually causes death within just a matter of minutes. It's nasty stuff. And protozoans, like for example, the one that had the little blue dashed line there, it is the Euglena. Euglenas are what we call an example of organisms that are mixotrophic. If you're mixotrophic, you can engage in photosynthesis, and you can engage in processes that aren't photosynthetic. So Euglena can be photosynthesizers or autotrophs, and they can also be heterotrophs like you and I. Diversity, right? Same thing with, well, I'll get to the purple bacteria in a second. However, on the other hand, if what you're oxidizing is not organic compounds, but something that's inorganic, you are still considered a chemo or a chemoheterotroph. Like, for example, the bacteria Sulfolobus. Sulfolobus oxidizes sulfur. Right now, it's a two-step process. It oxidizes sulfur, and then it uses the energy from the oxidation of sulfur to oxidize an organic compound. So it's got a step in between us. You know, we eat food and we oxidize that organic compound for energy. These guys oxidize sulfur, use the energy from that to then oxidize organic compounds for energy. And one that's really kind of cool is one they found living on the iron plates of the Titanic. It's a new species they discovered called Halomonas titantia, even named it after where they found it. And this stuff oxidizes iron. And the energy that's released from that, they use that energy to then oxidize organic compounds. So literally, prokaryotes can live in some very, very harsh environments and can digest some very unique types of food. These guys are basically living off the iron plates that form the hull of the Titanic. Now, one type of metabolic method is reserved for just a very select few group of organisms, and these are the organisms that are called photoheterotrophs. All right, so photoheterotrophs, like purple photosynthetic bacteria, use light as their form of energy and organic compounds as their form of carbon, as their carbon source. And that's the other dashed blue line showing you that many organisms can switch between two different forms. Purple photosynthetic bacteria can either photosynthesize, like you see way over here. They can also use light as a form of energy, but instead of using carbon dioxide, they can use other organic compounds. This is important for understanding the versatility, especially of prokaryotic organisms, Rhodobacter of prokaryotic organisms. And again, this goes back to the idea of the early Earth being a pretty harsh place for organisms to live. Excuse me. And the fact that we see these organisms that can live off of, off iron, sulfur, hydrogen sulfide, ammonia, right? These critters, could survive in areas of the world, especially in the early world and even today, where other critters wouldn't be able to make it at all. Now, if you're wondering what the little white thing is there right here, that's actually a plant. It's actually a flowering plant. It's not green though, so it doesn't photosynthesize. And we'll talk more when we get into plants about this type of plant. Actually, some growing on campus. I've been checking to see if they've come up lately. If they do, I'll show you some of them. But this is a flowering plant that doesn't photosynthesize. It actually lives off the roots or off the fungus that grows on the roots of other plants. So once again, what I would do if I were going to be studying for this is I would get rid of the pictures and I would just kind of concentrate on the different terminology and the different types of organisms that can carry out these metabolic processes. All right, what about oxygen? Well, we need oxygen, but not everybody does. We're an obligate aerobe, and many prokaryotes are obligate aerobes. Some are obligate anaerobes. In other words, they actually are poisoned by oxygen. You remember early on in the history of life, as the oxygen levels began to rise because of the cyanobacteria photosynthesizing, a lot of the prokaryotes were dying off. Oxygen, like I said, is a strong oxidizer. Obligate anaerobes then have to survive on engaging in fermentation or anaerobic respiration. And where would you find these critters today? Well, you wouldn't find them out in the open air, obviously, but you can find them down like mud at the bottom of the lake, near the bottom of the ocean. These areas can become very low in oxygen or actually have no oxygen. We call those kinds of areas that lack oxygen anoxic. So the bottom of the ocean, deep enough, right? Bottom of deep enough lakes can become anoxic, and you can find some obligate anaerobes living there. Facultative anaerobes have the best of both worlds. They can either live with or without oxygen. Nitrogen. Again, nitrogen fixation. To fix something in chemistry means to incorporate it into a chemical compound, and that's what nitrogen fixing bacteria can do. So again, the whole adage is, or the whole situation is, if plants need nitrogen, and we typically say that nitrogen is limiting to plants, when we talk in ecology about something that's limiting, what we mean by that is that which is in shortest supply. But again, there's plenty of atmospheric nitrogen. Plants can't use that. But nitrogen fixing bacteria will take atmospheric nitrogen and turn it into ammonia. From ammonia, they turn it into nitrites. Nitrates are then converted into nitrates, and that's what plants can then use. So that's very important. And some plants, for example, like legumes and beans, are very good at doing this. They're very good at fixing or incorporating atmospheric nitrogen. Plants use some of the nitrogen for their own use, but the bacteria that are fixing the nitrogen make an excess of nitrogen, and those nitrogen, that nitrates and those compounds are dumped into the soil, and they allow other plants to grow. That's what I was saying when we were talking about the evolution of life, that plants actually created opportunities for other organisms. So plants that have the ability to fix atmospheric nitrogen, or more specifically, plants that have the bacteria that live in them that allow them to fix atmospheric nitrogen, would have enriched the soil with nitrogen. And then, of course, there are other bacteria, as I said, that break down nitrogenous compounds, right? So we have this complete cycle. Organic nitrogenous compounds come from the decay of plants, from the decay of animals, and then those are recycled back to nitrates and nitrites, and denitrifying bacteria turn that back into atmospheric nitrogen. So from an ecosystem point of view, from a plant health point of view, prokaryotes are extremely important, aren't they? right? They're important for bringing that nitrogen so that these organisms can survive. Many prokaryotic organisms engage in a sort of a metabolic cooperation. Like, for example, that picture there, that's Anabaena. Anabaena is a filamentous prokaryote. Each one of those things in a chain there is a cell. And what I mean by metabolic cooperation is there are two types of cells. There are the photosynthetic cells that engage in photosynthesis, carbon fixation, right, to take carbon dioxide and incorporate it in organic compounds. And then the heterocysts are involved in nitrogen fixation. So it's the heterocysts in the Anabaena that do this process, take the atmospheric nitrogen and turn it into a material necessary for the Anabaena to grow. Biofilms, tooth plaque. You don't brush your teeth and you get plaque on your teeth. That's a biofilm, and a biofilm is again, it's a colony of prokaryotes, not a tissue, but tissue-ish, tissue adjacent, tissue-like, if you will. And in the blue there, you can see the yellow's teeth, tooth enamel, and the blue is a biofilm that's spread all over the top of that tooth. Streptococcus is involved, very often involved in creating tooth plaque. And the problem with having tooth plaque is that's where cavities come from, because Streptococcus ferments sugar anaerobically. When it does, if you remember from Bio 1, you produce acid, right? Lactic acid, and the acid erodes the tooth enamel. And that's where we get cavities from. That's why it's important to brush your teeth, because it removes the biofilm. All right, now it's time to talk about different types, diversity. 10,000 or more species, could be a lot higher, could be 10 times higher. We're not sure. We haven't named all the different prokaryotes out there. Most of the prokaryotes that we know about, again, are the ones that cause us problems. Early classification was centered on things that we talked about early on in the semester, easy things like whether they could photosynthesize or not, whether they could move or not. Morphology, right? Whether they were unicellular or filamentous, like the Anabaena, the types of spores they made, and then this last one, which is, of course, very artificial, how important they are to us as human pathogens. Right? We classify them that way. But recently, and I've mentioned this before as well, classification is going to be based on DNA, right? Either looking at the sequence of the amino acids, which you remember from Bio 1, the sequence of amino acids is tied to your DNA. Looking at the actual nucleic acid hybridization, right? We did talk about this earlier too, where they basically take two species with their DNA, right, and they split the DNA in half on both of them. So this is from species 1, and this is the base sticking out from species 2, and see how much they match up. The more they match up, the more base pairing occurs, the more they match up, the more closely related they are. And then sequencing the entire genome. In other words, finding out the entire genetic sequence of organisms, right? So that's, you know, and as I said, that's what we have with modern technology, the ability to actually look at the DNA. I'm going to point out a couple things before we actually get into the prokaryotes. So these are all the prokaryotes, these two domains, archaea and bacteria. And this is the domain we belong to, the domain eukarya. Notice among the prokaryotic domains, which one we're more closely related to. If you take a look at this, remember from what we did in chapter 26 and talk about relationships, you can see that it appears from the evidence we have that we're more closely related to the archaea domain of prokaryotes than we are to the other bacteria. Which is kind of interesting because the archaean domain of prokaryotes is, as I said, contains a lot of the very extremophiles, those prokaryotes that can live in very extreme conditions. It appears that we evolved from some of those more extreme living type of archaeans. All right, table 27.2 is a good table for comparing the three different types of domains on some major characteristics. I really only need you to focus on the ones that I've indicated with the red arrows. First of all, the one that should be obvious to you, prokaryotes do not have a nuclear envelope, whether they're in the domain bacteria or archaea. Of course, we do. Peptidoglycan, bacteria in the domain bacteria, peptidoglycan, archaeans do not, eukaryotes do not. This is one of the bits of evidence that suggests we're more closely related to archaeans than bacteria. The lipids in the membranes, and I'll show you those lipids in just a second. Bacteria, we have unbranched hydrocarbons, same as us. Domains have branched hydrocarbons. Now, that has important implications for their membranes, which we'll get to in a moment. Introns, the integrals. Bacteria very rarely, if ever, have introns. Archaea have some, as well as eukarya. So there's another little bit of evidence about the relationship. And the ability to grow in temperatures greater than 100. That's what I was saying. It's kind of interesting about who we are related to. Most eukaryotes, all eukaryotes probably wouldn't be able to survive at temperatures above 100 degrees Celsius. That's boiling, folks. But some species of archaeans can. Yeah, the archaeans are the extremophiles. Let's look at this the cell membrane difference. All right, where was I? All right, the membrane lipids. Let's look at that for just a second between bacteria and archaea. All right, the plasma membrane made up of phospholipids and a bilayer. And if you look over here on the right, this little picture, this is what the phospholipid bilayer looks like in bacteria and eukarya. And this is the one you learned in Bio 1. Right? Those are phospholipids arranged in a bilayer with the hydrophilic heads facing out and the hydrophobic tails facing in. Okay. In the archaean, though, things are a little bit different. The first difference occurs in the bond that occurs between the glycerol and the lipid tails. All right. In the bacteria or the eukaryotic lipids, that's called an ester bond. But in the archaean lipids, it's called an ether bond. Now, there's a difference between those two based on chemistry, which I don't want to get into. But what I do want to show you is archaean lipids, the ether bond makes them much more resistant to hydrolysis, makes them much more resistant to being torn apart. So again, it's that idea that archaeans are tougher. They can deal with much more extreme conditions. Archaean lipids are branched, and they're connected to that glycerol by those ether linkages. Bacterial and eukaryotic lipids are branched, but they're connected together by ester linkages. Now, the other difference that's important is that archaean lipids form a tetraether layer. What we mean by that is, if you can look up here, it might actually help a little bit. In archaeans, the phospholipid tails, there's no gap in between. They form from one phosphate head to the other, and they also are attracted to each other. It basically, if we looked at the membrane from the side, it forms layers. This is a monolayer. And that monolayer makes that cell membrane very, very durable. That's what allows the extremophiles of the archaeans to survive. They have a much more durable phospholipid bilayer than we do. All right, we got a couple minutes. Let me go through the archaeans. I won't be able to get through all of them, but we can at least get into some of the archaeans. The domain archaea, a lot of times, are the extremophiles. Three big groups of archaeans are the methanogens, the halophiles, and then the thermophiles. The methanogens are those that I was talking about that oxidize carbon dioxide, or they use energy by using carbon dioxide to oxidize hydrogen, producing methane. You find those oftentimes in swamps. You walk around in swamps sometimes, and if you smell, when you're walking in the mud, you smell that methane smell that's coming up. That's the methane from these methanogens. Many of them are obligate anaerobes, so they're going to live down in the mud of that swamp where oxygen can't get to them. These are also ones that are very important for herbivore digestion, living in the gut of things like cows, which is going to be very low in oxygen. You remember, those bacteria allow the cows to survive on cellulose, right? The cow itself can't break up the cellulose, just like we can't, but the bacteria in their guts can. Halophiles are those that can live in really salty conditions, and they make some really beautiful pictures. This is a bunch of halophiles growing around a thermal vent, or growing around a thermal pool. And these things love very salty conditions, well beyond the saltiness of the ocean, 9 to 10 times saltier. Halo means salt, by the way, and phile is to love. Thermophiles can live in really warm conditions, like the ones we found down around the geothermal vents. They can live in temperatures close to boiling. Their optimum temperatures can be 60 to 80 degrees Celsius. And again, having those differences in their cell membrane, the monolayer that we were just talking about, that's one of the reasons they can deal with these very extreme conditions. All right, well, that's all I got time for today. We'll finish up with the domain bacteria when we get back together next week. Remember, you do have your exam on Monday, right? And your assignments are due, chapters 22 through 26. The exam will be in the lab room, so come on down there, and of course, it will start at 8 o'clock. You will be allowed to use your notes over the last 20 minutes of the exam as well. So I'm going to go ahead and stop the recording.